We can start.
Okay. Thanks a lot. So warm welcome to everyone. My name is Jeroen Scheerder, and I will try to manage in terms of do the chairing of this very important session of Jessica Samogin. She is expected to present her work, her PhD work today, to share it with us with regard to the main important results of her work. For that, she will have 30 minutes to present her work. So warm welcome. It's somewhat cloudy here in Belgium. I don't know exactly where you are, but nowadays, of course, all of us have to deal with this COVID-19 still. So it's a little bit weird to see you this way. But anyway, we may maybe be happy that we still can continue with this kind of very important celebrities and meetings. I will soon start with the introduction of the jury members, very important. So a kind of introduction. What else can you expect? We have a clear program. As I mentioned already, the first 30 minutes, it's up to the candidate to present her work, after which she will also be invited to present what we call appositions, 3 appositions, after which it's the floor is open to the jury members to raise questions, and in total that will take, let's say, 1 hour approximately. After 1 hour and a half in total, we withdraw with the jury and have to deliberate, and I guess this will take usually 15 up to 20 minutes. So please do not run away from this meeting. Of course, you can relax, maybe drink something or go to the toilet, whatever. And I will make clear at what time we expect you to come back for a very, very important moment, the proclamation, but also the laudatio by the supervisor. And the last word, of course, is to the new, hopefully, doctor in biomedical sciences. But that's for later on. So then you have at least a clear idea about what you might expect. I would also kindly invite all of you, and I'm really looking at the people in the room, in this digital room, the audience. If you feel that you have a question towards the candidate, please feel free to ask it after the jury members. So but I mentioned it already because then you can prepare things. If you want to raise a question in another language, no problem. We can try to translate it. If you want to raise a question in French, Dutch, whatever, we can make sure to translate it into English. We will try at least. But that's a clear invitation to all of you, and I really want to invite you to, let's say, at least have 1-2 questions from the audience. So I will first start with the introduction of the jury members, and I first start with a special colleague, the external jury member, Professor Elisa Magosso, is an associate professor of the biomedical engineering department at the University of Bologna, Italy. The research of Professor Elisa Magosso, her research activity mainly concerns the development of advanced feature extraction techniques for biological signals and the mathematical modeling of complex physiological systems with the aim of understanding mechanisms of impairment in pathological conditions. Many thanks and a warm welcome, Elisa, Professor Magosso, for being with us.
Thank you. Thank you very much.
The second examiner, jury member, is Professor Patrick Dupont. Professor Dupont is a full professor of the Faculty of Medicine, program director of the Medical Imaging track, and chair of the Department of Neurosciences. With his studies, Professor Dupont investigates the functional structure of large-scale brain networks using several imaging techniques with the aim of understanding pathology-specific abnormalities. The third examiner is Professor Stefan Swinnen. Professor Swinnen is a full professor of at the Faculty of Movement and Rehabilitation Sciences and the head of the Movement Control and Neuroplasticity Research Group at our faculty in Leuven. Professor Swinnen investigates how aging affects brain structure and function and also studies the relation between movement, physical movement, and brain activity and connectivity as measured by different imaging techniques. The fourth and last, but not the least, examiner, Professor Kaat Allaerts, is an associate professor at our faculty, the Faculty of Movement and Rehabilitation Sciences in Leuven, and also the head of the Neuromodulation Laboratory at the KU Leuven. The research of Professor Allarts focuses on neuromodulation in healthy individuals and patients, primarily using non-invasive brain stimulation techniques and EEG-based neurofeedback. And then lastly, the promoter, the supervisor of this thesis is Professor Dante Mantini. He is a professor at the Faculty of Movement and Rehabilitation Sciences of the KU Leuven, and lead scientist at the San Camillo Research Hospital in Venice, Italy. The research of Professor Dante Mantini mainly focuses on the study of movement, perception and cognition using advanced brain imaging techniques such as high-density EEG. So that's what all of the jury members and the promoter concerns. I again repeat that we kindly invite you for preparing a question if you feel to do so. That is an opportunity that you maybe can grasp with both of your hands. And then I now want to give the floor to the candidate, Doctoranda Jessica Samogin. Please, the word is up to you.
Thank you, Professor. So, as you mentioned in the first part of this meeting, I'm gonna explain you what I've been doing for the past 4 years. My work could be summarized with the title you see here. It's long, it's complicated, so I'm gonna break it up for you and explain the meaning of each part. Let's start with the human brain, which is the organ that controls and regulates the functioning of the rest of our body. This is possible because of thousands of neurons, the starry things you see in the middle of the slide, that communicate together to process signals and properly respond to internal and external stimuli. The unit of their language is called action potential. Sequences of these action potentials are the neuronal signals. Let me just do one thing quickly, because I forgot to share the presentation.
Indeed, we cannot see the presentation, Jessica.
Yeah. Here. Here you are.
Okay, fine. Continue, please.
So, brain, neurons, action potential. So the language, so to say, the sentences of the neurons are the neuronal signals, sequences of action potentials. Depending on the features of these neuronal signals, we can classify them in 5 main brain rhythms: delta, theta, alpha, beta, and gamma. You might be able to see in the center of the slide that the signals corresponding to these rhythms differ quite a bit, also visually. But most importantly, they differ because they are mostly associated to certain behavioral conditions: sleeping, moving, studying, meditating, and so on. And in the past years, they have also been associated with different cortical regions. The task, so to say, that for me is the most interesting, is what is technically not even a task, is the resting state, which corresponds to the mind-wandering condition. You are supposed to be relaxed and let your mind free to wander and think at whatever you want to. In the experimental setup to measure that is basically putting a person sitting in front of a screen, fixating across this presented on that screen, and asking this person to relax and think at everything. It doesn't matter what. Just not to focus on anything in particular, which is easy to say, but not so easy to do. What we want to understand is what the brain does when you're doing that, so when you're just thinking. To measure brain activity in that condition, we have 3 main types of acquisition systems: functional magnetic resonance imaging, magnetoencephalography, and electroencephalography. The first one is probably the most known among common people because it's often found in hospitals, and you might have done an MR scan for whatever reasons in different parts of your body. It's good, this acquisition system, because it provides images, the black and white ones, with good spatial resolution, meaning you can really see a lot of details of our brain. But unfortunately, it does not measure direct brain activity. It has low temporal resolution and it requires a dedicated room, and the scanner itself is quite big. On the other hand, instead, MEG and EEG both directly measure brain activity, either the magnetic or the electric part of it. They have good temporal resolution, and the images that we can reconstruct from them have acceptable spatial resolution. Still not as good as the fMRI ones, but definitely good. MEG still has a cumbersome equipment, whereas EEG consists basically in a cuff with a lot of electrodes and wires coming out of it. This cuff goes onto your head, and a laptop and an amplifier provide to acquire the data. Because of all these advantages, we decided to continue using EEG and try to promote the use of EEG data. Once we have EEG data, what can we do? What do we want to measure? We want to measure functional connectivity, meaning if we have our brain, we select some regions, we measure its activity with one of the three devices that I mentioned before, and we try to understand whether there is a relation, a functional relation between these regions. The mathematical methods to do so can be classified into main families. One is based on the similarity of envelopes, either time envelopes or power envelopes, meaning how the green curves look, how similar they are. The other big group of connectivity metrics is based on the phase, either synchrony or phase lag, so kind of a constant phase difference between signals. Envelope correlations can be applied on any kind of data, fMRI, EEG, or MEG. That's why we went for this method. We decided to use this method. Time correlations on fMRI data have given in the past this kind of result, this kind of images. They are called connectivity maps, and they represent resting state networks. So the yellow, orangish regions within each small head are cortical regions that are frequently found to be active together in response to the same kind of stimulus. You might be able to read the names of these networks, but the names tend to be related to the type of stimulus. So there's an auditory network, a visual network, sensory motor, and so on. So we have EEG data acquired during resting state and envelope correlation to measure the functional connectivity between brain regions. Which kind of questions do we want to answer? What is still missing? Or what's still missing 4 years ago? There were some methodological issues and knowledge gaps. In particular, EEG data cannot be used as they are once they are acquired. They need to be processed, and the processing steps are a lot, and that might be complicated. So we wanted to create an automated pipeline that processed this data and reconstructed cortical activity. EEG is collected on the scalp. We want to know what happens on the cortical surface. As for the knowledge gaps, while most of the connectivity studies done in the past were based on fMRI data, which cannot give us information about the frequency. That's why we wanted to know more about the role of frequency on functional connectivity. And we wanted to know whether that role changes or has an impact on connectivity within one network and between networks. And lastly, whether changes in resting state connectivity can be related to behavioral performances. So the main goal, you might have understood that, is investigating frequency specificity of the interactions within and between networks during resting state. To do so, we created 4 different studies. The first one aimed at developing the pipeline, and the following 3 aimed at analyzing frequency specificity in the interactions within one network, between several networks, and finally studying the effect of aging on functional connectivity and the relation between this connectivity and motor performances. Pipeline. Well, it's a pipeline analyzing EEG data, so guess what? We have high-density EEG data as input. High density means the acquisition system has at least 64 or possibly more electrodes. that cover the head, the scalp. These raw data, as they are called, are processed with different steps in order to obtain cleaned channel data. Cleaned meaning they contain mostly or possibly only neuronal activity. Then we also use individual MR scans and electrode positions in order to create an individualized head model, which is a mathematical model describing the positions of the cortical sources and how how these sources can create the activity we measure on the scalp. Indeed, clean channel data and in the individual head model are the input for the source reconstruction, which reconstructs source activity. Sources are basically, you can imagine those as groups of neurons on the cortex. Source activity is decomposed in the time frequency domain in order to perform connectivity analysis. Connectivity analysis give usually as output what you see here, which is called connectivity map. For this first study, we decided to apply ICA connectivity to test the goodness of our processing and reconstruction steps, source reconstruction steps. The ICA is a data-driven method that decomposes the signals, the data, into statistically independent components, either in time or in space, meaning either the time courses are independent or the connectivity maps are independent. The results we obtained are the ones you see here. If you are familiar with connectivity maps, you will recognize the networks as we know them from fMRI data, which is kind of our golden standard, our reference. So nice, the pipeline works. It allows us to perform frequency-specific analysis, meaning we can filter the EEG data or consider the EEG data only within some frequency ranges. However, we do not know the connectivity profile of these networks, how the connectivity strength changes in different frequencies. That's why, for the following studies, we decided to move on to the seed-based connectivity approach. In this case, you really select certain cortical regions that are called seed, nodes. Well, I will use both interchangeably, and we measure activity of these sources. So again, we measure scalp activity and reconstruct it at the cortical level, and we try to understand whether there is or not such a functional connection between them. We are using resting state data, so we're not focusing on the default mode network. The default mode is known to be basically the hallmark of the resting brain, in the sense that it's the only resting state network that is active during rest and deactivates during task. All the others behave the opposite way. The conditions, the mental conditions that activate the network are usually self-relevant thoughts, inward-directed thoughts, memory retrieving, mental imagery. And this activates the four core regions of the default mode network differently. These four regions correspond to the yellow dots on the brain that you see here, and are usually subdivided into two subsystems because the mental conditions activate these subsystems differently. There is a midline core involving the posterior PCC and the anterior MPFC regions, and there's a medial temporal system with the left and right angular gyruses, which works sometimes independently. And finally, as for the frequency signature of this network, meaning the frequencies that are usually found to be associated with its activity, we know that alpha and beta band play a major role. So if we use these four nodes and we apply our pipeline on the EEG data and we try to construct seed-based connectivity maps in different frequency bands, we will have this kind of results. In this case, the seed is the MPFC. And as you can see, the maps corresponding to different frequency bands are quite different, meaning not all of the regions are present in all of the maps, and even those that are always present change color in the maps, which corresponds to a change in the strength of the connectivity between the MPFC and that particular region. If we repeat this analysis on all of our seeds, we will have the results you see here, which explains how, or which shows how only in the alpha band we are able to see the 4 regions of the DMN, the 4 main regions of the DMN, regardless of the seed we have chosen. So in all the 4 maps on the right side, you will see the DMN as we know it, and you can compare it to the same kind of connectivity maps reconstructed from fMRI data with the same seeds. So we're starting to realize that the frequency does play a role in connectivity, in functional connectivity, because in different frequency bands we have different connectivity maps. However, how much is the influence of frequency on the connectivity between these regions? We define within network connectivity as connectivity, functional connectivity between nodes of the same network. In the table you see at the bottom of the slide, the rows correspond to all the possible interactions between the DMN nodes. The columns are all the frequencies between 1 and 80 Hertz, and each dot represents a frequency for which the interaction between the 2 nodes in its same row are statistically significant. The interaction is statistically significant. The first results that pops up is that only in the interval between 9 and 11 Hertz, we have 1 dot for each row, for each connection. So only in this interval, all the functional interactions are statistically significant. Indeed, if we select, let's say, the PCC as node for our analysis, for our connectivity map, and we reconstruct that map in this at 10 Hertz, actually, we are able to see all the other regions of the DMN within the red circles. If we instead select a different frequency, for example, 14 Hertz, and we keep PCC as seed of our analysis, the only interaction that involves PCC at 14 Hertz that is statistically significant, meaning there is a dot for that interaction on the in the table, is the one with the anterior region, MPFC. Indeed, in the connectivity map, you will see only you will see some red parts only in the area corresponding to the MPFC. In a similar way, 17 Hertz, all the interactions with PCC are significant. That's why we see that all the other regions, left and right angular gyruses, and the MPFC, the frontal one, even though not all the possible interactions are significant at 17 Hertz. So to summarize all of this, we say that, yes, frequency has a role in functional connections within the DMN. In particular, there is a frequency interval between 9 and 11 Hertz for which all the interactions are significant, and there are other frequencies, lower and higher, that support mediate functional connections between the regions depending on the regions involved. This is true for the DMN, and it's one network. But you may know that there are several resting state networks in our brain. And in order to accomplish a lot of tasks, we need this network to cooperate together, to work together. This is measured by the between-network connectivity, connectivity between nodes of different networks. In particular, previous fMRI studies have found that connectivity within each network is always higher than connectivity between the networks. We can use EEG data, we can have information and use the information about the frequency, so we wonder whether this is true, the results that we have from fMRI, if that's true in all frequency bands or only for some. And in case it is only for some, it's the same frequency band for all the networks, or does it change? To do so, we selected 5 other resting state networks for a total of 21 seeds, being spread out throughout the cortex. And we reconstructed connectivity matrices that you see here. In order to understand this figure, you need to know that within network values, connectivity values are on the diagonal. So on the diagonal, there is the average connectivity within the network itself. On the lower and upper triangular matrices, there are the between networks connectivity values. The connectivity, which represents the connectivity between, for example, the nodes of the motor network and the dorsal attention network. So you can see, just by watching these 5 matrices, that they don't really look alike one each other. They are quite different. In particular, for each row, the value on the diagonal is not always the highest of that row, meaning is not always the most red one, the darkest one. And it varies depending on the frequency band in this case. So to measure how much different is connectivity on the diagonal compared to the one outside of the diagonal in the same row for all the networks and all the frequency bands, we need to understand this new figure. So red squares represent connectivity within the network that's higher than that's between the networks. Blue squares, the other way around. So connectivity between the networks higher than connectivity within the network. And the stars indicate results that are statistically significant. So what we know from fMRI, meaning within network connectivity, stronger than between network connectivity, it is true also with EEG data. However, it depends on the network and on the frequency band. It's not true in all frequency bands. In particular, for bilateral network, we have this effect on alpha and beta bands, whereas for lateralized networks, so networks whose main regions are on one hemisphere, only one hemisphere, they mostly use gamma. This effect, is found in gamma bands. All of what I've shown so far is true for young adults, young and healthy adults. What about older people? The aging affects our body and our brain in several ways: structural, biochemical, functional. And again, most of the studies done so far used fMRI data. So again, we don't really know what happens in different frequency bands. We want to understand that. We want to understand how aging affects the functional connectivity pattern. And also, we want to see whether this possibly different connectivity pattern relates or not to the behavior, the bimanual coordination of the participants, motor performance of the participants. Bimanual coordination is a commonly used task where the participants are holding 2 dials and are rotating them in order to move the red dots along the blue line, following or trying to follow the white dot. It might look simple for a young mind. It will probably look quite difficult to an older brain. So first, connectivity and aging. So again, we have an hypothesis we wanted to test, which is called the de-differentiation hypothesis, according to which in older brains, connectivity between networks is higher than that in younger brains in the same condition. And this could correspond to a lower level of segregation, meaning usually the networks are very specialized, cortical regions in general are very specialized, and each region does a certain task, certain job. They might need to cooperate, they might also be able to accomplish a task by themselves. Lower segregation means, or could mean, they need to cooperate more in order to get to the same result. So we reconstruct connectivity matrices, again, for young and old separately. And in particular, I would like you to look at the position of the white stars. They are basically in the same place for the same networks, meaning that the pattern of within network connectivity stronger than between network connectivity is mostly preserved with aging. The big exception is the DMN, but we know that in older adults, the connectivity within the DMN is quite disrupted, so this result is kind of expected. But as for the other networks, the stars are basically the same. You could almost switch the two images, and you wouldn't be able to say, OK, this is younger and this is older adults. So the functional segregation seems to be preserved with aging. However, if we compare the connectivity matrices, frequency band by frequency band, we see these results. The blue squares represent conditions in which connectivity is stronger in older adults compared to younger adults. The red squares is the other way around. And you see that the blue squares involve, especially those with the star in it, are mostly outside the diagonal, meaning they involve between network connectivity rather than within network connectivity. So again, the hypothesis coming from fMRI data, it is true. However, it's true only for some networks in some frequency bands, not for all networks and all frequency bands. And what about the relation between connectivity at rest and motor performance? In particular, motor performance, OK, motor network. Connectivity involving the motor network, either within the network itself or with the other networks we were analyzing. And as for motor performance, while we were looking at bimanual coordination, so bimanual, the level of bimanual coordination, or also called bimanual synchronization. What you see here is are correlations. Correlations between connectivity at rest and motor performance in young and older adults separately for all the different frequency bands. The stars are only in for the older adults and only in the beta band, which is interesting because beta is the band or rhythm that is mostly associated with movement in general. And it's also interesting that it seems that the stronger the connectivity within or between motor network, the better the performance in this particular motor task. So if we try to explore a little bit more these results, because we used more than just one network, we will see this. Again, these are correlations. And in particular, you can see that the effect of higher connectivity corresponds to better motor performance is true, particularly for those networks that are task relevant, as the attention networks, dorsal and ventral attention, and the motor system, and also the default mode, in beta band and in beta band only. So we might be able to relate connectivity at rest and task performance and know it beforehand, because this, remember, that is connectivity at rest, not during the task. So to summarize all of this and put together our results, we say that, yes, frequency plays a role in the functional connectivity between resting state networks. And in particular, nodes within the same network use oscillations at specific frequencies to interact with each other, depending on the regions that are necessary or that are activated in that particular condition. The same applies for interactions between networks. Within and between networks, our connectivity is dependent on the frequency. And finally, this connectivity, which is specific in the frequency domain and also in the spatial domain, meaning it's specific to certain networks, relates to behavioral performances. All very nice, however, there are limitations in my study. Limitations due to the techniques, the methods I've used. In particular, well, except for the first study, I used seed-based connectivity, which, of course, depends on the choice of the seed. We always try to select seeds within the main regions of the networks. However, if we move a little bit that seed, the connectivity pattern could change, particularly if we select a region or a point that is shared between networks or that has connections with multiple regions. And of course, the choice of the seeds means deciding a set of coordinates in a template space that then has to be adapted to the individual head. So it's tricky, but we need to define some seeds in order to be able to test our hypothesis. As for the frequencies, I used the predefined frequency bands in order to be able to compare my results across groups and across participants. It could have been interesting using individualized frequency bands, particularly for the older adults. However, that would have not allowed me to compare results across participants. My young and older adults were not the same people acquired at different time points of their life, were different people, so I could not just make a comparison one by one. That's why I needed to define frequency bands. And for similar reasons, I could only compare results at group level, which gave me some statistical power, yes, but also did not allow me to compare the same condition, resting state, in the same subjects. And particularly for the EEG and fMRI data of the second study, that would have been really interesting, since there are techniques to measure EEG and fMRI activity at the same time. It wasn't possible at that moment of my PhD. And finally, as I said at the beginning, our pipeline tries to create an individual connectivity results. And this because of course, individual EEG data, but particularly for an individualized head model, which uses the MR scan of the subject, which uses the electropositions of the subject, but unfortunately could not include the information about individual conductivity values, mostly because we couldn't measure them. The one that we used are parameters which are often found in literature. However, the variability of these conductivity values could potentially change our results, at least a little bit. And the more individualized is the head model, the better, if you want to really know what happens in one person's head. Therefore, what could we do in the future? While some of these limitations could be, of course, overcome with future studies, more subjects, and so on, but there are other venues that we can explore, starting from what I've done so far. From a methodological point of view, dynamic connectivity. So what I used in all of my study was the whole time course of the signals and consider it as a single piece, so to say, and try to understand which was the average connectivity behavior, functional connectivity behavior of my networks. However, with dynamic connectivity, we analyze the signals with shorter time windows, and we are able to identify also networks that are transient, that don't appear often, or that appear for a short time interval. So of course, we could start answering questions like, do the amounts of time that this network spends during rest change with aging? Do the frequency of these changes change with aging? And so on. Directed, or also called effective connectivity. In all my studies, I just looked at the existence of a functional connection between regions. Directed connectivity instead looks at the direction of this connection, so it tries to understand which regions leads the activation of the other. And finally, phase-based metrics. If you remember, at the beginning I said that connectivity metrics can be subdivided in two main categories, two main families. One is the one I used, and is based on correlations of envelopes. And we know now that it gives good results also with EEG. What about phase metrics? They measure and they use a different feature of the neuronal signal. So they could possibly give us information about different connectivity structures that are based, again, on the phase synchrony or phase lag, rather than just correlations of power or correlations of envelope of time courses. And finally, experimental options. Resting state, of course, it's easy to use, particularly in with not healthy people in general, because it doesn't require much effort. You are just supposed to relax and think. It shouldn't be difficult to misunderstand what you are asked to do. But of course, we can also look at connectivity during task. We know the connectivity profile between pairs of regions during rest. We wonder, how does it change with a task? Does it change depending on the difficulty level of the task, for example, depending on the age of the patient? How can we modulate that?
Jessica, can you please finish up?
Yeah, give me just 1 minute, basically. As for the second interesting task condition is sleep. We sleep, our brain luckily does not sleep. However, the main oscillations, main rhythms that dominate the brain during sleep are in the theta and delta bands, which are lower compared to alpha, beta, or gamma. So how can networks that usually during wake work with higher frequencies still work and actually do stuff during sleep with lower frequencies? And finally, clinical applications. It would be interesting trying to understand if there are markers, biomarkers that predict or characterize certain neurological disorders. And maybe if we can try to develop targeted intervention to maintain, correct, preserve the brain of these people. So try to, for example, slow down the degeneration and so on. And that was it. Thank you.
Okay, thanks a lot, Jessica, for the clearly structured presentation. I would now like to invite you to present and present the 3 expositions you prepared.
Yeah, right. Yep. So, my oppositions are that any efficient system is based on cooperation, organization, and specialization. From our whole body down to the single cell, each part knows what to do. Together, they accomplish even the most difficult tasks. We should learn from the nature. Secondly, doing research can easily become a 24/7 job. Researchers should be encouraged to make time for anything that helps distracting and relaxing. It will improve the efficiency during working hours. Instead of living for working, we should work for living to do both of them better. And the third one, AP value is thought to represent the quality of a study. However, it depends on factors that cannot always be controlled for. The reproducibility of the results and the correctness of the procedure that led to them should be more important than AP value.
Okay, thanks a lot for this. Then we now switch to the part of the examination, let's say. And I first pass the word to Professor Elisa Magosso, who has 20 minutes at maximum for her questions. Please, Professor Magosso, take the floor.
Thank you. Can you hear me?
Yes.
Clearly? Okay. Thank you, Jessica, for your so well-organized and stimulating presentation. I have some questions for you. A couple of them are about methodology, specifically about source activity reconstruction to then compute connectivity. We know that source activity reconstruction methods provide a current dipole for each voxel of the source space. So we have 3 time-dependent components, but connectivity analysis requires just one single time series to be derived for each voxel. And so fixing dipole orientation. There is not a definite solution on this in literature. And different solutions indeed are adopted. One of them is to use the orientation orthogonal to the cortex surface at that voxel, so to obtain a signal which is representative of activity in dendrites of pyramidal neurons. The other solution, which is the one you adopted, is to use the component maximizing the projected variance.
Yeah.
Which seems less more linked with biophysics of neurons. Can you motivate your choice and provide some specific advantages linked to this choice? What do you think about that?
Well, thank you for the question. Well, indeed, we, at the beginning, were using the component that was orthogonal to the cortex, so the standard method, so to say. And then we realized why not trying to use the components that maximizes the variance, meaning the PCA of these three components. First, empirical results gave us basically the same results. So using one or the other did not affect our results, our connectivity analysis. And this, I would say, was the main reason for us to continue with that venue. But secondly, there's also a computational advantage, at least with our pipeline, because we can calculate the components that are going to be used, so the components maximizing the variance before getting to the source reconstruction, to the connectivity step itself. It's just faster in terms of computational time. Also, if I think I have three components, and I want to keep only one of them, I would use, or at least try to use, the one that explains most of the variance in my data, which would be like the exponents, the most important in my data. So yes, I will not have always the same direction of my vector, which is that I have with the standard method. It's not always the same, but it's always orthogonal to the cortex. But I will use the one that explains the most the data, the dipole activity itself.
So you are assuming that the two components that you are neglecting, the other two components among the three principal components, are essentially noise.
No, maybe not completely noise, but the part of information that they carry is very much smaller than the main component, the first component.
Yes, I think that also the other methods try to do the same. So because it assumes that the orthogonal, the projection of the dipole along the orthogonal direction is the one carrying most information related to communication among neurons.
Well, indeed, neurons are basically orthogonal to the cortex, so it makes sense thinking that's the components that should be retained, at least for communication, is the one orthogonal to the cortex. Yeah, physically, biophysically, it makes sense.
Yes, biophysically, yes. But so you are saying me that results did not change substantially using the two methods, is correct?
Yeah. We tested it thoroughly because we wanted to be sure. It was a new technique, and we didn't want to make mistakes.
Okay, so I think this is an interesting result because there is still controversy, so I think this may be also good to know for other researchers that there is not so big difference in using one or the other solution.
Yeah.
Okay, thank you for your answer. The second point, still on source reconstruction, is about individualized realistic head models. You used realistic head models in your pipeline, but we know that in many cases we do not have magnetic resonance images of subjects. Have you tested your results using an anatomical template or not?
Not me personally. What we do is if the MR is not available, we don't use the individual electrode positions. So we use basically the template head. And with the template head, I have collaborated in other studies using that one because, again, the MR was not available to many patients or to all the MRs, and still good results were obtained also in those papers with those collaborations. So it's not the best option, I would say. If it's possible to use individual scans, it's better using them. But
okay,
yeah.
Maybe so. Maybe there are other aspects that are more important for an accurate reconstruction. For example, to have an appropriate number of electrodes or to use the correct methods for inverse problem solution. Maybe these are factors more important than using a realistic head model.
There are a lot of factors, actually. Of course, if I think at what was available, for example, at universities in Italy, even in a research center, it was not definitely on high-density EEG montage. So we had to accept lower data with lower spatial resolution rather than, thinking 32 channels, which is not high density, which is actually used at the hospital. So that's not good, not so worse because it gives results. Source reconstruction, it is a big problem, methodological problem. We still don't have the perfect solution. Because of the nature of the problem itself, we are trying to use the best one available at this moment. But of course, it's also the best one depending on the scope of the research itself. Do I want to know what's going on in the head of my patient or my subject? Do I want to know whether there is activity in this particular region or there is not? So I think it's a combination of all the different factors. And what we did is testing realistic head models, but created with different methods. So different segmentation methods, for example, or different co-registration of the electrode methods, or the method that creates the mathematical model itself. To test, at least in that, how much accuracy we need, or we can hope to have with the head model. But of course, it's not the only thing, or possibly not the main thing that plays a role in understanding EEG activity.
Okay, thank you. I move on results and their interpretation. So first question is about study 3 in this presentation. So in particular about the comparison between within and between connectivity. I think it's a key result. You found that the frequency range for which within a connectivity is higher than between a connectivity depends on the specific network. So for example, ventral attention network and language network show higher within connectivity in a gamma band. This is specific for these two networks. And you interpreted these results in terms of nodes distribution. So you said that bilateral networks tend to exhibit higher connectivity within network connectivity in alpha beta, while unilateral networks in gamma band. I think this interpretation may be valid, but I wonder if other interpretations are possible, can be attempted. For example, you mentioned the thalamus as an important structure for alpha generation, but you did not mention the hippocampus that could have a big role in gamma generation and could be strongly linked with the nodes in those networks, ventral attention and language attention networks. There are other interpretations you think are possible that can enlarge the the actual interpretation, the current interpretation you have proposed.
Yeah, I agree with you, actually. Well, as for the thalamus or for the hippocampus, for the thalamus, we can't really say how much its connectivity pattern influences that of the cortical networks, because we try to analyze the activity, the thalamus activity with the EEG. However, there are so many nodes, and the spatial resolution of EEG is not good enough to allow us to really meaningfully study each single nucleus. And therefore say, okay, it's because we see gamma band in this unilateral network because of that particular node of the thalamus. Hippocampal activity, it's too deep, and yes, for us to measure it. So again, we can't really say much about it, about its influence on the connections with this unilateral network. However, structural connectivity is known to predict, up to a certain point, functional connectivity. So definitely the structural connections within the head could explain why we see gamma in the unilateral networks and we don't see gamma in the bilateral ones, for example. So there might be it is possible that connectivity within these networks is higher in that band, not only because their nodes are closer together than those of the default mode, for example, but also because they both communicate or interact with an external region, a certain other region, which influences or which determines the activity in gamma. So that's possible. It would have been better knowing the structural pattern, for example, of each of my subjects. Again, these data were not available, the DTI or DYI, for example, to make assumptions or to conclude something about it. The only thing we saw was, the main thing we saw was, okay, unilateral networks have this within, higher than between network connectivity in gamma, and it's not the case for the other ones.
Yes, this is true, but maybe that could be maybe there is a relationship between the preferred, let's say, oscillations for a given network, even because of the specific role of that network.
Also.
So, for example, gamma has, we know that gamma band, gamma oscillations has a specific role different from the alpha oscillations and for the beta. So it may also be that there is a preference for a rhythm which is functionally more relevant for that network.
Yeah, indeed, it could be also that hypothesis. I was studying data at rest, connectivity at rest. So it could be also, let's say, motor network, beta bands. Activity in the motor network is usually correlated or related with beta bands. However, this between relation was not significant or so much significance in beta bands for the study you mentioned, the third one. The significant relation between the two types of connectivity was in alpha. For ventral attention, while gamma band is known to be related to attention, state condition where the participants need to really focus on something. So that would make sense, even though it was just resting state. Let's say the usual language that these regions talk is in gamma. When they are active, therefore they might still use the same language when they are at rest. Same for language network itself. Yeah.
Okay. Maybe I have still some time for the last question.
We still have 3 minutes, Professor Michelson.
Okay. So very quickly, just to see if I have correctly understood our results on older subjects. An interpretation better. because it's still uncertain to me the relationship between the differentiation hypothesis and the compensatory mechanism you have mentioned, not in this presentation, but in the manuscript, Jessica. Because it seems that there is your result is that motor performance is positively correlated with beta band connectivity, especially in somatomotor and dorsal attention network, both intra-network and inter-network connectivity. So what I understand is that subject with reduced segregation in beta band exhibits higher motor performances. So it seems that the differentiation is a sort of compensatory mechanism, like as if a network asks for help to other network to perform a given task. Is this interpretation correct, or there's something not that I didn't understood?
Well, technically, the differentiation and compensation are different, meaning the differentiation describes just higher inter-network connectivity, which does not have a behaviorally relevant function. It's just more noise, more connection in the brain. Whereas the compensation hypothesis says that these higher connections, this higher connectivity, plays a role in, for example, executing a motor task. So the higher connectivity, the last result, higher connectivity in beta band for better performance would be a compensatory mechanism, or could be interpreted as a compensatory mechanism, which should compensate for possibly the structural changes that appear in the brain. Or I like to think at it as somehow as the gamma for the attention network, same idea. So when an older person has to do a certain movement, a certain task, motor task, for sure, attention network and motor network need to be to work together, possibly more than in a young person. And since this happens or should happen recurrently, every time this person does a motor task, in a similar way as for the resting state networks, whose pattern is detectable both during rest and during task, these new interactions, these new connections, which are created during the task, will then at a certain point be seen also during rest. So it's like creating the root by just using it in order to. This is my interpretation.
Okay,
and that would fall within the compensation hypothesis, compensatory mechanism.
Thank you. I think I have finished my time. Probably I'm a little bit late. Thank you very much, Jessica.
Thank you.
Okay, thanks a lot, Professor Magosso. Well timed. And now pass the word to Professor Patrick Dupont. Please, Professor Dupont.
Okay, Jessica, I enjoyed your presentation, but of course, we have to challenge you a bit. And I would like to discuss a little bit resting state networks. So can you elaborate a little bit on the reproducibility of the resting state networks defined by fMRI or EEG, and actually how this reproducibility is assessed?
Okay, well, thank you for the question. Resting state networks, as I said at the beginning, it's not a task. Resting state, it's not a task. And when you are mind-wandering, you can think at really whatever you want. And these thoughts could activate networks in a different way, depending not only on the subject, but also within the same subject. If I ask you now to rest for 3 minutes and I ask the same question this evening, probably your thoughts will be different. So it might be strange that we are able to see these networks in different participants. Different conditions, with different acquisition systems or data. This is possible because, at least with the envelope correlations, we are looking at what happens in the brain on average. So over 5 minutes, of course, the longer the acquisition, the better, I would say. I tried with 3 minutes and it works. With 5 minutes, it's even better. So the longer the acquisition, the more time these networks have to be active, to appear in the connectivity and the acquisition. And we average over them. It's like taking pictures of the brain and checking, okay, how many times do I see the default mode? Okay, 8 times. Default mode is here. This allows us to compensate for the high intra and inter-subject variability, because we
but if you talk about, for instance, the default mode network.
Well, that's easy. It's the network of the resting state.
Yeah, that's true. But then I'm just wondering, like, you mentioned this actually in your text somewhere. You choose basically the 4 main nodes of this resting state network.
Yeah.
There are actually many more which are described, but they are clearly more variable, and I understand why you choose the more robust ones. But then I'm just wondering, even the robust ones, how robust are they among subjects? And not only subjects, but also among the different techniques that you use.
That's what statistical analysis tell us, how robust they are. They are robust according to our threshold in the statistical analysis. If so, we are able to see those at a group level. If I take my 19 participants and I check their connectivity maps, I will probably see different regions, more regions of the DMN. For some participants, there will not be even the 4 ones, maybe only 3 or 2. But at group level, and the more subjects, hopefully the better, we are able to see, for sure, the main regions. Again, they are the biggest one, the most recurrent for the DMN as well as for the other networks. And this is true basically for all the acquisition systems. The acquisition systems tells us which kind of information we take from the brain, but the activity underlying it is the same. And especially for MEG and EEG, they are basically measuring the same thing, because magnetic and electric fields are related. And that thing, that what goes on in the brain during resting state, is across subjects more or less reliable. Of course, if it wasn't reliable, we wouldn't have been able to see anything, not even the default modes during rest. It's easier during task. During task, you can lock the activity to one stimulus and reduce the noise. During rest, no. That's why we need to be sure that EEG activity, in my case, is really only EEG activity and not noise once we get to the connectivity analysis.
Yeah, but that's partly driven because you selected actually the major nodes, let's say, of these resting state networks.
Yeah.
I'm just wondering what would have happened if you would actually include also the other nodes.
Well, it depends if they are measurable with EEG. I mean, if a node is deeper or it's in the cerebellum of the DMN as well as any other network, I want to make sure that I can actually measure its activity. Only 4 nodes. Well, there's also another constraint with EEG, which might not apply to DMN, but to other networks. Yes, we can't use nodes that are too close to each other in space. So their position on the cortex is too close. Otherwise, we basically end up measuring the same activity because...
True, that's something you mentioned in your presentation as, I think you called it, that EEG had acceptable resolution.
Yeah, it's not fMRI resolution, but still, it's not that bad, particularly for high-density systems. Low density, well, it's debatable how good the resolution are. But again, it depends, as I said to Professor Magosso, on which is your research question.
Absolutely, yeah. But just to elaborate a little bit further, you also mentioned at some point that rest measurements, certainly in patients, are easy. You don't need to have specific instructions or do some task which may for us maybe relatively easy, but for some patients might be even at that point too difficult. So I certainly see clearly the advantages. But then on the other hand, I think you mentioned a few times, I think, yeah, resting state is actually a state of kind of mind wandering. And that's actually a better description than resting state, because the brain is actually never at rest.
Yeah, indeed.
So I like your point of view in that sense. But then at the same time, yeah, it's also very difficult to control, of course. How would you do that?
Well, you basically can't, and I've experienced it myself as a participant in several experiments. That's, well, already the fact that you're supposed with EEG as much as with fMRI to stay still, either sitting or laying down, looking at a cross, trying not to blink, and looking at a cross might seem easy, but then you're focused on the cross, and you're also focused on not thinking at anything in particular. So yeah, I know it's complicated. You can't really control it. You hope that the person is doing what you ask for. Same with mental imagery, for example. How can you really know that the person is imagining what you asked that person to imagine? And it's doing that and not thinking at other stuff, or doing the complete task and not stopping at the beginning. To control for that, I think you need participants. Of course, you can check whether these people, the activity, brain activity or power content of the EEG is in line with what you expect. That's for the resting state and mind wandering. Each of us do something different.
One thing you obviously can do is, I mean, you actually alluded already on this, the way you instruct subjects is very important. If you would say you are not allowed to think about anything, yeah, that would trigger me and I cannot do it anymore. On the other hand, what some people and groups do is after a resting state measurement, they actually have a kind of a short interview or a questionnaire to see what kind of things actually happened during that short period. So that at least you have an indirect, it's not a full control obviously, and you rely on what people tell you, but at least you have some idea of what might have gone through their mind. So they typically classify this in subdivisions. Okay, but let me move to another question, which I was also a bit intrigued by. At the end of your thesis, you write something about the clinical applications, and you mention, and now I quote, the results presented in this thesis suggest that high-density EEG, high-density-based connectivity may be used in clinical populations to identify novel disease-specific biomarkers.
Yeah.
Can you elaborate a little bit on this, and how would you actually assess a disease-specific biomarker that can be used in a clinical setting? So that actually would mean that you need to make a statement about an individual patient.
Well, I would first start trying to classify selecting a disease, getting as many patients as I can, and try to understand whether there is something that all of these patients have in common in terms of functional connections between networks and frequencies or frequency bands. If I can make assumptions or have a priori hypothesis about the regions that might be affected by that disease or the frequencies, then I will try to focus mostly on those, because of course I made my life easier by looking at networks and frequency bands. I could have done the same, just looking at each single frequency and applying a brain parcellation, meaning way more regions than the few, let's say, that I studied. So I would try to control the regions and the frequencies, get my patients, and identify something that's could be specific of that disease, possibly even comparing it with what happens in healthy population. So my results once they will be reproduced on a way larger scale, could tell the researchers in general, okay, see, normal brain, healthy brain, the motor network has higher connectivity in the alpha during rest rather than in beta band, or language network in gamma, for example. I have a population of patients, same disease, and then I find out that the language network does not have higher connectivity in gamma band, but in delta band, and maybe at 3 Hz and not 2 or 1. That would be a biomarker. And then the next step would be trying to understand whether we can use that biomarker to predict the onset of the disease, or if that is related only, I mean, if it appears only once the disease is there, so once it's too late. Or if the disease is somehow treatable, even with stimulation, for example, or training, can we bring that biomarker to a normal, the thing that is a biomarker, to a normal condition by training the participants, by treating it somehow or not? That would be my idea. And with EEG, because it's just smaller and easier acquiring an EEG than, for example, an MR scan at rest without tasks, because, as I said before, it's just easier and less, there are less misunderstandings if a person is required to, let's say, do nothing with proper instructions, but do nothing, rather than doing a task or also feed neurofeedback, which I tried. It's not so easy, actually. So
yeah, but I think it's also important to, if you want to bring it at some point to a clinical setting, is that we actually also study reproducibility of your results, and that reproducibility can be on the short run or longer run, but also some sort of robustness. Like if I would choose slightly different seeds, as you alluded in your limitations already, if you have slightly different seeds, your results should hopefully still be robust enough. If the quality of the data is a little bit less, it also should be robust enough if you want to use it in a clinical setting, obviously.
Yeah, indeed. My results, as they are now, are the first results, so to say. They need to be validated over and over again on bigger population, for example, between the third and the fourth study, I had data acquired with different acquisition systems, so different number of electrodes, different people, not different number of people, and also the length of the acquisition was shorter in the last experiment. So that was a first step toward validating the results. Then, of course, there will be applying maybe ICA again to check the networks are still there. Am I seeing something wrong? Rather than with ICA, maybe we can, I'm not saying that only with seed-based we can find the biomarker. ICA could be good, for example, maybe even easier, because you don't have a priori hypothesis in that case. But just, I'm trying to promote the use of EEG, basically.
I'm not sure if I still have time to ask a short question.
Well, a very, very short one, because we are already over time.
Okay. It actually relates to your third opposition. Because when I was looking at this, I was a bit intrigued, because you mentioned that a p-value is thought to represent the quality of a study. And actually, that's what I always learned, that a p-value is something different and has nothing to do with the quality of the study. So can you elaborate a little bit how you come to this statement?
Well, in my experience, I found that journals, in particular, always ask for results with statistical tests, which threshold is below that p-value, basically the threshold p-value. So p-value below 0.05 or lower. And that's what I mean with quality of the test. So they don't, or most of them don't really look at, are your data validated? Is your pipeline validated? Can you reproduce your results? Just they look at a p-value, where I put the stars on my plot, which for me is not enough, is not it is not the main thing that tells you whether my research, my study is good enough to be published or not. It might be that I wasn't lucky and I had a cohort of people with a lot of variability, so I see a trend there. I cannot acquire more data for whatever reason, and the publisher says, no, but your results are not good because it's not, the p-value is not below that threshold. Yeah, okay, but then look at all the rest.
I disagree a bit, because some journals do accept also negative studies, but of course there is always the issue of power of a study. Yeah? But that would be a discussion which would bring us too far, so I will stop here and we can go to the next jury member. Thank you.
Thank you.
Thanks a lot, Professor Dupont. And then I now switch to Professor Swinne. Please, Stefan Swinne, take the floor.
Thank you, Chairman. Congratulations, Jessica. This is a nice piece of experimental work. What I particularly am excited about is that you've tried to push the boundaries of current methodology for the study of these complex brain networks. And of course, this work is also much aligned with the ambitions of your promoter, and I would like to commend him for this supervision of your work. A few questions. I will first start with a question on these short range and long range connections that you occasionally talk about. And they are quite interesting to the extent that you also make a link with the different frequency bands, in the sense that the long range connections are probably more mediated by the lower frequency bands and different for the short range connections. So how important is this short versus long range connectivity for you in relation to the study of brain networks?
Well, for me, long range, short range is related to the physics underneath the neural communication. Low frequency waves, low frequency oscillations, mediate long-range communication, mostly because the physical properties of low-frequency signals allow them to travel for a longer distance. And the other way around, short-range, higher frequencies. However, structural connectivity is a base for functional connectivity, but it's not, they don't overlap completely. So the relation between the frequency of the oscillations and the position of the nodes over the cortex, yes, it is important. It's not the only factor. However, it's, at least in my studies and for me. So what also Professor Magosto said before, it's not necessarily that language network has higher connectivity in gamma, within network connectivity in gamma, because the nodes are closer together compared to those of the other networks, bilateral networks, for example. So
does that also have any implications when you look into interhemispheric connectivity versus intrahemispheric? I mean, if let's say these interhemispheric connections are more long range, would that also imply that the lower frequency band may play a more prominent role in interhemispheric connections and interhemispheric interactions?
Since the information needs to pass from one hemisphere to the other, they also need to go through the connection between the hemispheres. So possibly lower frequencies because longer range. I mean, it's not that the neural signals just travel through the air. They need a physical pathway to travel on. So longer connections, lower frequencies. Although, according to my results, the lower frequencies are alpha and beta band, which are definitely not the lowest possible in the range of interesting frequencies for brain activity.
But isn't it also important to consider actually the structural strength of the connection? For example, if I think about the connection between Leuven and Brussels, I could be concerned about the distance between these two, but maybe I should be more concerned about how many lanes I have to go from Leuven to Brussels. And so the latter would rather, I guess, refer to the structural connectivity in the sense of density of connections, number of connections we have, maybe rather than the distance of these connections?
I think it's a combination of both. Yes, it's also the distance, but if I have a lot of ways to get to Brussels or a wider highway, so possibly more information can travel through that connection. It could be, well, it might be that it's easier to communicate for that node, but it could also be that that node is particularly busy. So I would say it's a compromise between distance and strength of the connection. The strength of the connection would be more important for the within-network connectivity, or as we saw for the older adults, well, again, according to my interpretation, the bigger connections, the more stronger one between motor and attention systems could lead to better motor performance because, the highway is bigger or has been created and then it's becoming bigger.
Okay, so you already elaborated on this potential link between functional connectivity on the one hand and structural connectivity on the other hand. And of course, we know you can also have strong functional connectivity in the absence of direct structural connectivity. Yeah. But my question is, what do you expect from the context of EEG work about the potential mapping between structural connectivity, white matter microstructure as a scaffold for functional connections? What are your hypotheses in that respect? And particularly then in terms of, or in relation to frequency bands, would you expect a good mapping between integrity of structure on the one hand and connectivity, functional connectivity on the other hand?
Well, a lot of studies have been done about this relation between structural and functional connectivity. So I would expect that there is this relation is somehow preserved also with the EEG data. It's not that connectivity based on EEG has nothing to do with the structural connections. It has to do with it. Possibly, I would maybe say the correspondence could be higher where when it comes to connectivity within the network and the frequency bands for which this connectivity is stronger. So again, also the relation between structural and functional could depend on the frequencies. Maybe not as much as connectivity per se, but still could change because connections at, let's say, not so important frequencies could be less strong. The path maybe is not there yet, could be mediated by different nodes, so it doesn't maybe require a clear anatomical path to travel on. Whereas connectivity that's commonly used, so connectivity within the network or corresponding to bigger highways, bigger paths, yes, it would. I think it will correspond to the structural connectivity, or it can be predicted by structural connectivity.
So maybe we should move on to the different frequency bands. I wonder how evolution has arrived at a kind of architecture, brain architecture, in which these different frequency bands that you have identified play a prominent role to manage to mediate the interactions between brain regions. Why is it the way it is? It's almost an existential question, I guess.
Yeah. Well, I didn't create the human body, so I have no idea why. I mean, I don't know for sure why is that. I can hypothesize, of course, on that. I can just elaborate on that. Well, one of the most important things for me is that different regions do not disturb each other while they are working. There is no crosstalk. So I would say specific frequencies for specific networks or for specific processes, behavioral condition, whatever, are necessary to avoid crosstalk, avoid awakening regions that are not necessary for that goal. And that's why or for me, that's why there are different frequency bands and different networks. Different frequency bands for different networks. Then the frequency of the oscillations, of course, influences also the speed with which oscillation travels. Can the information go to one point to the other quickly? Does it take longer time because the wave is slower, because there are more steps to get to the other region? And that would have an impact at a behavioral level. Do I need to react fast or not? And then, of course, different regions, depending probably on their anatomy, are associated with different frequencies. I mean, the alpha band and the resting state are just one thing. Why is that? It's not clear yet. It could be that there's a common generator, thalamic generator, that basically leads all the functional connections in the alpha band. It could be that all the networks kind of relax at that frequency, the networks that are not the DMN, since the DMN in resting state is working.
So would you argue that the lower frequency bands are more critical for integration in the brain, and the higher frequency bands are more critical for functional specialization?
Well, since the high frequency short range, if we stick to that, then short range means local activity, and it depends on the definition itself of short range, but then it could be related to segregation.
Okay, thank you. Maybe moving to a different type of question, that this is about the link between EEG and fMRI. So you've made a few critical statements about fMRI, and I fully agree with you. And the Bolt signal is a very indirect proxy of what you could consider neural activity. But I was a bit disappointed when we started using more and more EEG in terms of understanding what these signals mean for brain activity in general. For example, if you look into power analyses and you study changes as a result of learning, or you study differences between groups, young versus old, or stroke versus controls, if you think about brain activity, the Bolt response, it's easy to identify whether you have lower or higher degrees of brain activity. But in terms of EEG and the use of power, it seems extremely complicated to make a link between power and brain activity and neural activity. And particularly then, this is even more complicated because the interpretation may also differ across different frequency bands. What are your thoughts about that?
Well, I would not exclude fMRI from as a imaging technique. Even though I'm in favor of EEG, I would use the two of them, possibly even combined, depending on what you want to look at. So if you want to look at the power, and with fMRI we have good results, and you don't want to know the frequency specificity of that power, or you don't, you're not looking at how power changes in short time windows that are below the time resolution of the fMRI, then why not using fMRI? It's there, it's available, so yes. For DEG, I think it's more complicated because, while there is a lot of information there, also the fact that we have frequencies, we have frequency bands, is just another degree of variability, another problem or thing to think at, to try to fix and solve. EEG power, yes, we use it. I used it also in the last paper, and indeed I didn't really find anything particularly meaningful or easy to understand, to interpret, only with the power. Power and motor score, behavioral score, scores were not related. Power didn't tell us anything about how good or bad a person could perform. Connectivity could. So maybe the potential use of EEG would be higher in the functional connectivity part rather than in the power analysis, power studies. As much as we cannot use EEG for structural connectivity. So of course I need the scanner to do that, to study that.
Okay, thank you. I guess my time is up. If not, I have one more.
Well, it is Professor Swinner, so I have to pass the word to the last examiner. Apologize for that. Thanks a lot. Kaat Allarts, please.
Okay, thank you, Chairman. Hi, Jessica. Good afternoon. First of all, I would like to follow my colleagues, and first and foremost, of course, I'd like to congratulate you with this very impressive line of work that you presented. Also, the presentation today, I thought it was really extremely clear and comprehensive, so I really liked it a lot. Thank you for that. It was also a real pleasure for me to go over your work, and it really sparked my interests in many ways, and therefore I also prepared some questions to go over some points that actually, maybe sparked my interest the most, and that I would like to learn more about.
Okay, thank you.
Okay, so first question actually relates to your second chapter, where you report that the frequency-dependent connectivity profile between pairs of the default mode network, that these mostly reside around a peak at the 9 to 11 hertz frequency band, and you call it also in your presentation the carrier frequency band. Now, of course, as you know, this is found in the alpha band, and we know that this is most prominent during resting state. And in this respect, I was actually wondering whether the identification of this band during rest, and then the identification of this band as the most prominent one for defining the connectivity profile, whether this indeed relates to the fact that you measure during rest, and that it might therefore not be only specific to the default mode network, but that rather for any resting state network, that you will find this connectivity profile, and that this will be merely the case because other frequency bands might not be as prominently identified compared to the alpha. And I think if I looked at some of your figures, it indeed showed that the alpha band was indeed most prominently identified. And I was wondering, before you start looking into the connectivity profiles or the correlation between these power fluctuations, whether you think it would be necessary or relevant to first set some kind of threshold, and in order to establish whether indeed there were reliable power changes in each of these bands, the frequency bands, and that sufficient power is actually needed to have reliable connectivity patterns. Otherwise, that you could maybe look at correlations between noise in the different frequencies.
Okay, so well, as you said, alpha band is the band of the resting state. Mm-hmm. That's clear. And the default mode network is the band of the resting state, which is the relation between the two, meaning what I saw was just noise due to the resting state condition and therefore to the alpha band, or not. Well, I hope not, in the sense that it was this result was obtained on a group of subjects, actually on two groups. I mean, for the first study and then the following one. So the more times I can replicate that, the more reliable it will be. If it is influenced by the alpha, we actually performed a power regression on, it was another study, not the first one, the second. So we calculated correlation, connectivity. Then we calculate the power of these networks in all the frequency bands and regress that power out of the connectivity values. And connectivity did not change. So the power effect, or if you want to say the effect of noise, on that connectivity result was neglectable, was not there, basically, because connectivity did not change when we removed the power information from the values. The sub-interval, the small interval, 9-11 hertz, might be specific to that group of people. Could be wider, could be not, and this is related also to the definition of frequency bands, to the fact that I used the same frequency bands for all the subjects, and also the same definition of the frequencies for the same subjects. If we look at different intervals, or we try to individualize for example, the alpha peak, then we might see that in a person, these 9-11 hertz could shift to, let's say, 8-10 or 8-9 interval, and so on. But I don't think that what we see is just because it's alpha, it's resting state, and therefore the functional connections are significant in that range. In terms of power, yes, there is a clear alpha peak in all the networks, but there are also smaller peaks in the beta bands for sure the motor networks that I remember, and I think also for the dorsal attention network, meaning there is more than just alpha, even at rest, in these networks, in terms of power, and also in terms of frequency, actually. Because it's true that it's all calculated on the same data, but in alpha band, with that frequency range, we can reconstruct the network for the first study, the ones on DDMN. So we can reconstruct the network using that frequency. We cannot reconstruct the whole network using another frequency, depending on the seed. The similarity of the connectivity maps between fMRI and EEG were the highest in that frequency band. So there are a lot of factors that put together suggest that it's not just noise or result get by chance.
So alpha was for many of the networks the main driver, but not solely the alpha band.
Not solely the alpha band, and also alpha band. Okay. I mean, there could be like the thalamus driving the alpha band activity, but then networks are working together in other frequencies. It's like it could be a background noise.
Okay. Thank you for your answer. Maybe we can go to the next question. So there, I have actually a question about something you wrote in your introduction and later came back to in your discussion. So you indicate that the static representation of resting state networks, as observed using the more slow bold signal using MRI, that actually that you might reveal a richer structure when once analyzed in time and frequency, as done with a high definition EEG. And so this prompted me indeed to think about something that you then mentioned in the discussion, namely that, indeed in the field of MRI research, there is this increasing shift towards the characterization of also dynamic functional connectivity, rather of what is now called the static connectivity, as you also showed in your future directions in the presentation. And indeed, hereby it is acknowledged that any brain state when it would be identified, that it presumably only lasts for a couple of milliseconds and then it might change. But could you comment how you think that your present approach can actually also would actually also allow assessing connectivity in resting state networks in a more dynamic fashion? Well, and what would be, in your opinion, also the time resolution there?
Well, the easiest way would be to shorten the time windows. So instead of using 3 minutes, 5 minutes of resting state, going down to 10 seconds or even less, we have milliseconds information activity. Usually the frequency acquisition frequency of EEG data is around 1000 Hertz. So we have a lot of samples. How short or how long the windows could be, well, that can depend on what you want to look at. So, of course, you can go down again to a few seconds. I didn't test it yet. For sure, since the time resolution of EEG is higher than that of the functional fMRI, we can use shorter windows, time windows, than what is now used with fMRI. Just because, the signal they measure is like a delayed version of the neural activity. So there is a lower limit on the length of this time window. And then for EEG, with my approach, shorter time windows will be the first easy step to start with.
Yeah, because indeed, maybe a related question in the BOLT response. Yeah, functional connectivity studies assessing these spontaneous fluctuations in the BOLT response, they are usually filtered around the signal around 0.1 hertz. And yeah, I was actually wondering, in your approach, you actually also look at more fluctuations, fast and slow fluctuations in the power of, for example, the alpha rhythm, and not actually the raw signal. So I was wondering, do you have any sense of what the frequency actually is for these fluctuations in power in, for example, of the alpha rhythm? Because that would probably determine the resolution of any dynamic window that you can adopt.
Yeah, I will need to have at least one period of my fluctuation within the time window. Alpha, beta, or gamma, or lower determines the length of the window. So if the period is short, meaning frequency is high, I can have shorter time windows. And the other way around for slower frequencies, longer time windows. Yes, as a minimum length.
But does it still relate to the frequency of the band that you assess? Because if you look at the power fluctuations, it's a different signal, no?
It's power envelopes. Yes, it's a slower version of the signal because you take the envelope, so you connect together all the peaks, basically. If you don't want to use band-limited power, then you can, let's say you want to use just time correlation, so correlation between time courses, which is what is done with fMRI, then you have a different resolution. So actually using time courses would allow us to use shorter time windows, even a more dynamic approach, dynamic in the sense that the time windows are shorter and therefore higher in number within the same time, within the same acquisition.
Okay.
So with power, we slow it down a little bit.
Yeah, indeed. Okay. Maybe one final question in the interest of time, maybe?
We have 1 minute still.
Okay, I will then go to my last question, final question. It's more of a general question. Which part of your thesis did you actually like the most yourself? Or do you think that would have had the most impact? And could you also elaborate why you chose that part or that result?
Well, the last one. The first study on the aging, because I've always liked to acquire data on people and do something that's meaningful and possibly helpful for the population. Aging is something that affects all of us at one point, fortunately or unfortunately, depending on how you see it, and understanding what happens in the brain of my grandma, for example, at least at a population level, at a group level, for me is extremely interesting. And also maybe trying for myself, when I will be older, trying to realize, okay, I cannot do that because of this problem in my brain. I can try to avoid having this problem if I train or stuff like that. For me, is the most interesting. And that's why also I would like to move to patients, real patients. So aging for me is not a disease, it's just something that happens, but still is a condition that it does not correspond to the healthy young brain. And even that, what is healthy? Maybe an older person is still healthy, it has some problems, but then from a cognitive point of view, it's perfectly normal. So because it's something that's not so standard, so to say, as a healthy adult, for me the aging was the most interesting, definitely.
Okay.
Just because I couldn't
Thank you very much.
have other people. Thank you.
Okay, thanks a lot to all of the jury members. Although it seems that we needed somewhat more time for the jury members' questions, I would still like to give the opportunity to the audience to pose a question. Is there someone from the audience? I don't know, Jessica, whether it's possible technically because I have I cannot see who's in the audience and who wants to raise a question. But if someone feels the need to do so, please take the opportunity.
They are hiding themselves like students at school when the professor is going to interview them. So I would say no. I don't have questions. Do you have, guys? No. They say no.
There's no questions. Okay. Then I would like to thank all of you already, and please again do not run away. You can have a break. We will withdraw with the jury, let's say for 15 up to maximum 20 minutes, I guess. So at the latest 2:10, I guess we will be back. But I would invite the jury members to switch from to another channel so that we can have the deliberation. Thanks already, and see you within, let's say, 20 minutes.
Thank you. Sì, rimango in live stream. Tanto sono tutti italiani che mi stanno a guardare più Simon, quindi. Lo capisco, mi spiace. Mi dispiace che sei beccata tutte le domande, più che altro. Io ti guardavo e stavo per ridere, tutto strozzo, Moro, raga, mo. Devo parlare. Ma grazie a Dio che ho fatto lo studio con te. Ascolta, come vado a spiegare? Vabbè, ho sputato. No, perché quello che ho fatto io poi l'hai usato anche lei nella sua, quindi... No, ma a parte quello, Marti non usa headmod, cioè usa tutto template e non posso...
Vabbè, io ho preparato in bagno prima che la faccio qui.
Cazzo che ne ho bevuto.
Guarda che il bagno ma sono stata qua milioni di volte già.
Aspetta.
Però Federico mi ha detto che
È giù in fondo. Gente in Italia, vi avviso solo che dalle 2:10 si comincia. No, vabbè, adesso potete riposarvi e poi tornate alle 2:10, grazie. Tu ridi, ma non hanno capito quello che dicono. Non è colpa.
hanno capito, no? Che noi siamo obbligati a dire
Sì, ma non hanno... Certo che camicetta bianca e mascherina, no? Tu sei viva, mia madre dice, no? Perché quei tipi là di... Quei tipi... Mia nonna questa. Quei tipi là di oggi gli conviene tenerti là a lavorare perché da te hanno solo da imparare. No, non è
lo sentirà con 30 secondi di ritardo forse, ma Marco Marino dice fa dei complimenti da parte mia a voce.
Grazie Marco.
Marco Marino fa i complimenti alla signora Jessica Samohin a voce. Che bello anche sentire Agnese che va in bagno. Io ti tengo aggiornata su quello che...
Oddio, c'era anche Jordan, porco. Non ho capito niente perché ho parlato inglese. Jordan, il moroso della Lisa.
Ciao Lisa.
Vuoi venire qua? No, temevo peggio, cioè voi mi avete calmato. Giuro. Vabbè, aspetta, che hai detto agli italiani? Ma hanno chiesto che hai detto? Non ho capito, Fede, quando è finito sto meeting? Sì? Possiamo andare anche sulla terrazza se volete, se il vento non ci porta via. Raga.
Vai, rapida.
Vabbè, gli dice, leggi tu i ringraziamenti e ti do a posto mio, ok?
Sì, dammi un po'. Allora camminetta. Così andiamo. Che fai? Scusate.
fasciata te?
No, quanto fa con lei?
Se vieni qua non fa freddo.
No,
No, io sei già un caldo.
no, perché non voglio entrare in visio.
Sei già in,
Visual.
sei in Eurovisione.
Dov'è la carina? Peccato che non hai potuto fare la reception, perché se la facevi qua poi era nella sala del
Ma era
castello,
strabello.
lì era bellissimo.
Ho
Ti
visto.
stanno facendo la tenda della reception fuori.
No, è che sarebbe stato troppo complicato portare su la famiglia, test contro test, e dove li metti, dove li metti. Tanto cara, tanto. Però sì, sarebbe stata bella anche la location, perché alla fine qui è piccolino, ma fuori da
La
Dio.
sala non è
No, il problema è il proiettore, cioè voi non avete visto praticamente niente. Madonna, se lo rifate. Cioè, se vieni,
Hai fatto i colori?
ma non solo.
di distanza.
Ma tanto siamo io, te e io, tesoro. Aspetta.
Dio, che già fa freddo fuori, poi fa freddo anche dentro.
Volete una cosa?
Ti
ricordi
la
questo?
position?
Sono un po' più fell.
Cioè, queste io le ho rifatte, adesso sono grosse, sono 1.5 o
grosse.
addirittura, no, quasi 2 di Matlock. Ed è così. Quelle
Beh,
prima
si
erano
vedeva.
molto leggere.
Si vedevano, anche quella... Anche quelle dove poi le robe sparivano.
Sì, tipo, no, vabbè, quella con i cosi, questa.
Mm.
Si vedeva anche il grigio.
Io qui non lo vedevo, ad esempio.
No, si vedeva.
Quindi io da qua non so
Cioè,
cosa sto
bisognava
vedendo.
sapere forse che c'era del grigio.
Vabbè. Scusa, ho freddo adesso.
No, veramente.
Vabbè, comunque...
No, aspetta, stai nuda intanto che facciamo la foto, poi vabbè, lo so.
Ma tanto dobbiamo aspettare il discorso. Vuoi 2 foto?
Lei
Così, dove ti sei vestita per niente?
mi sono vestita per niente. Dai, fai foto. Ma questo sta male però, non me lo tolgo.
Ma no, tanto lo so dopo quando facciamo le foto al dottore.
Madonna,
Dopo le
schifo.
facciamo fuori.
Sì, che mi scaldo un attimo.
Comunque, come alla ripresa di maggio, cioè soprattutto muovono le mani, soprattutto muovono le mani.
Ah, comunque tutto questo sta venendo registrato, sappiate.
Sì.
Ale.
No, metti su una slide quelle di prima e fai finta di parlare così. Volevo farteli parlare col titolo,
Ma scusa se le facciamo dopo? Perché tanto poi...
Vabbè, comunque, il fatto è che avevi la linea. Cioè, sembrava un'aureola.
Lo so.
Sposta sta testa.
Lo so, però non...
E se no ti metti poi dopo
Sopra, proprio. Mi avanti. Ah, mi aveva detto il tizio, pensa quanto ho capito. Sai quello dell'altra volta che era venuto
Sì, l'ho
mercoledì?
visto, ancora me la sa.
Quello è Steven. Vuoi? No, ce n'era un altro che era qui, che mercoledì scorso ci dava una mano, mi ha spiegato come fare. Mi fa, ma no, ma quello lì ti serve il microfono, anzi questo prima di tutto, perché quello che ti introduce vorrà parlare da là. Da là. No, non hai capito. Quello che mi introduce sta a casa. Vabbè, allora il pubblico vorrà fare quello lì per parlare, farti domande. E no, alla fine mi fa, ma chi è che viene di pubblico? Io dico, guardi, c'è il numoroso.
Per la cronaca io avevo fatto una domanda, ma sono stata interrotta.
Sì, c'era lui che era così tipo.
Questo PC
Cazzo.
è...
Sta da fare. Comunque non so cosa aspettarmi adesso, cosa devono dirmi.
serve a niente.
Ma no, ma ho detto
Poi ti
che...
mettono la
Vabbè, adesso ho freddo. Ho detto...
ti chiamano
Jessica Samochin.
universale.
Io che mi preoccupo di imparare il nome dei tizi e poi Jessica Samochin. Mica mio città. No, Scherder. Jerun. Lui, Jerun. Vabbè, anche quelli della palestra mi chiamavano Samo e mi guardavano. Zin. Se non è presente
Vabbè, quelle
Vabbè, Jessica, dai. Non so, non guardano mai film americani.
Vai, che ti interessate?
Mi sento molto guardata. No, prima è quello.
Carlo, concentrato. Beh, sono fatte
quella che hai fatto al posto mio, là al Begin Off, quella che l'avevi fatto di prova per me, al Begin Off con la mascherina. Cioè. Alla tua defense metti un cartonato, tanto sei fotogenica. E l'audio sotto che va. Ecco. Sei giusta. Cervello che gira. Minchia, 40 messaggi. Cioè, scusate, mica non posso dire queste cose. Ho realizzato... E sì che mi sono scritta tutto quanto, però se io avessi condiviso lo schermo quando l'altro stava ancora parlando, vedeva se stesso. Quindi ho pensato per lui... Beh, ma ad esempio, l'opzione 2, ad esempio, io mi sono beccata tutti i meeting, lo so, l'opzione 2, tutti i meeting che tipo guardi il computer, ma la webcam è da tutt'altra parte, ma almeno guarda la webcam. E poi... Sì, era... Ah, sì, è vero. Ok, opzione 3 è sempre così. Sì, ok, ho capito, i peli del naso magari no, Ma capisci perché non volevo mettere la webcam del mio computer, perché sennò mi vedevano da qua, cioè.
No, magari sai fare dei piccoli telecomandi.
Ma poi rimane... No, vabbè, quella presentazione è nero. Non ci vedo. Con la presentazione è nero e ci sta, ma con la Skype no, perché mi arriva tutta la... è chiaro. Devo leggere, non ci vedo.
Quindi c'è il grande discorso. Hai preparato i
Sì. Ho preparato il... Ah no, lo devi leggere tu? Devi leggerlo tu?
Scusa?
No, Dante mi ha detto il suo e quante parole erano, quante caratteri erano, per matchare un po' la cosa.
Ma questa position che però è una raccolta di cliché in cui volevi andarti a cercare.
Ma non devono essere legati a quello che tu hai fatto, perché la prima era inizialmente il cervello è fatto da tante parti che come lavorano tra loro E invece le nostre non possono. E quindi quella del cervello non ho dovuto cambiarla mettendo il sistema, la natura, una roba del genere.
Ma ieri era lì a niente.
No, ma può essere anche... Ti chiedono cose, sì, e puoi anche doverle spiegare, nel senso tipo con una slide, con un'immagine, con qualcosa di pratico. Le Madonne cadono dall'alto, a position 1.
Posizione aurea, no?
Ma tipo quella...
Io la pagina bianca con tutto in production.
Lavoro, quello è lavoro stress.
Hai scritto là? Sì, come leggere?
Abstract. Però ti chiedono anche cose sulla tesi, cioè manco mi ricordavo di cosa avevo scritto.
Finirla.
Questa domanda. Qua. È quella di Julio. Voleva fare una collaborazione tra me e Julio. Solo che Julio, organizzato con me, si è dimenticato. Sì, ma questo ancora 2 anni fa. Fa sì, no, perché facciamo? E poi... E lei è quella che fa autismo, che è stra interessante. Però sono tranne la Magosso, che è italiana a Bologna, Dupont è a medicina, è quello che ci ha messo 2 ore a cercare. Sì, lo so. Vabbè, ma non sto dicendo... Sì, ma non sto dicendo nulla di male. Suinen è il capo di Dante, quindi è da noi. Scherder lo stesso è da noi? Cap anche? Basta? Sì, ma l'idea di queste commissioni è del tipo quello che ci capisce tanto, quello che ci capisce un po' meno e via dicendo. E poi Gaia. Guarda che stavo per togliermi le scarpe e a un certo punto non ce la facevo più. No. Devo chiederti le ciabatte. Stanno tornando. Devo dare la comunicazione in Italia. Aia. Non mettetevi le cose. Il ciapinciolo dietro perché è scomodo. Come manca solo la mascherina, guarda. Sì, non ho abbastanza spazio dietro le orecchie. Ci vedo tutto proprio. Taglio la testa sto primo, scusa. Aspetta che ho perso le cose. Sì, no, sì. Cioè, devo farlo in inglese e ce la fa. No. Non so se posso, non sapendo se potevo, vabbè, glielo ripeto stasera. Quindi posso farlo in italiano?
Okay, welcome back to all of you. Very important moment for us, and I hope all of you can hear me. On behalf and under the authority of Professor Luxels, rector of the KU Leuven, considering that Jessica Samogin, born in Conegliano, Italy, on the 17th of November 1919, is a holder of the diploma of laurea magistrale in biomedical engineering, issued by the Università di Bologna in 2014, seeing the statement of the examining board appointed by the rector, from which it appears that Jessica Samogin has submitted and publicly defended a thesis on large-scale analysis of neuronal oscillations in the human brain during resting state, on the advice of the examining board and in accordance with the requirements of the decrees concerning higher education in Flanders, I hereby confer upon Jessica Samogin the degree of Doctor of Biomedical Sciences, Leuven, this 5th May of 2021. Happy to be the first one, Jessica, to address you like this, so congratulations, Dr. Samogin. Well done.
Thank you. Thank you.
I hope you will enjoy this special moment, this special day for you, and I now pass the word to your supervisor for the laudatio.
Thank you. Thank you, Chair. Dear Jessica, dear Dr. Samogin, congratulazioni. Congratulations. The goal you have set 4 years ago, obtaining a PhD at the KU Leuven, is finally achieved. I want to express to you my joy and satisfaction for this important result. In 2016, you had completed your master's studies in biomedical engineering at the University of Bologna in Italy. And at the time, I was looking for a PhD candidate and decided to be proactive and search for candidates with the right set of skills and with previous experience on a research topic. So after finding your profile on LinkedIn, I contacted you and asked to organize a video conference. I remember then that when we talked, I was strongly impressed by your drive and your positive attitude. So I was very happy that you have accepted my offer to start a PhD with me in the motor control and neuroplasticity research group in Leuven. As a biomedical engineer, you had the right background to conduct research in a complex research field, such as the large-scale analysis of brain networks using EEG. However, there were many steps to be taken to achieve the research goals, which were ambitious, but within your reach. During the PhD, you had to deepen your knowledge about brain structure, brain function, movement, and cognition. You have also learned how to work and communicate with researchers with different backgrounds, such as movement scientists, physiotherapists, psychologists, biologists working at our faculty. I'm happy that you decided to take on this challenge and you worked together with me in Leuven in a multidisciplinary and international environment. I'm very proud. I'm very proud about the quality of the work that you have carried out, which was published in excellent journals in brain imaging. You have developed important methodological solutions for the analysis of brain connectivity, and then you have applied them to better understand the impact of aging at the neural level. Your work will enable further connectivity studies based on high-density EEG in healthy participants, but also in neurological patients. At the personal level, you have shown to be strong and determined, to be prepared to address any difficulty encountered during your PhD studies. You have always been kind and dedicated time to students, collaborators, and all colleagues. During the last year, we have been affected by the COVID-19 pandemic, and as many others, we had to change the way we worked. Indeed, we have been unable to meet in person for several months, but we stayed in touch with the video conferences. I always found you calm and confident in these difficult times, which is something I truly admired. Your hard work and perseverance have led to an important results, the PhD degree, and it's finally the moment for you to be celebrated by your family, friends, as well as by the professors of the doctoral commission. I want to tell you that I consider your PhD very important, and I'm grateful for the trust you have given me when you accepted working with me 4 years ago. I'm also very happy that you decided to continue working at the KU Leuven for your postdoc. I look forward to the studies that we will be able to carry out together in the months and years to come. Once again, Jessica, I would like to express my warmest congratulations to you for obtaining this PhD. I'm certain that the experience that you have gained in the last years will enable you to achieve other important objectives in the future. Thank you.
Thank you, Dante. Grazie.
Well, then the time is now. Thanks, promoter, for this nice speech. For the final speech, we look at the new doctor, Jessica Samhagen. Please take the floor again.
Okay, so getting here has been quite a journey. Nonetheless, this is going to be the most difficult part of it. All started over 4 years ago, as Dante mentioned, thanks to the research position that Dante offered me. At that time, doing a PhD or moving abroad were my priorities. However, this project was too captivating to say no, and thankfully, I understood that before it was too late. Then I came here, and I felt like Alice in Wonderland. I've done all of my study in Italy, where the academic environment wasn't so idyllic because of several research opportunities and highly international environments that are available here, but definitely not there. And also from an architectural point of view, where the great cold buildings built among factories in the outskirts of the city, as in Cesena, for example, were replaced by parks, woods, and the river surrounding the Niger. Heidi had the goats saying hi to her in the famous cartoon, while we had squirrels right outside our office windows, coming and seeing us every day. So for all of this, I have to thank you, Dante, really. If it weren't for you, I wouldn't have experienced all of this. I wouldn't have gotten to this point. Also, you guided me through these years, created opportunities for me to collaborate with other labs. You can quite literally send me to conferences to present our work, but mainly to learn from others how we could make it better. Of course, even though you were my main supervisor, the professors of the committee that followed me in this journey gave us precious comments and insights, which allowed us to improve the studies that I've presented earlier this afternoon. I won't deny that before the first meeting we had back in 2017, I felt quite scared, whereas now I was curious to hear your point of view, besides being worried sick, of course. So thank you for supporting and teaching me many, many things. I will give it a try pronouncing your names. Pardon me if they don't come out as they are supposed to. Thanks to the professors Jeroen Scheerder, Kata Laarts, Patrick Dupont, and Stefan Svinen. And to Professor Elisa Magosso, who was the first to really introduce me to the magic word of EEG, neurons and signals processing, back in 2014. She was the supervisor of my master thesis, and thanks to her, I got where I am now. Thanks to Professor Svinen and Dante again, I'll be able to stick around for a little longer to explore more features of the aging brain during my, hopefully, postdoc. Work aside, at KU Leuven, I got a chance to meet many interesting people with whom thoughts and experiences have been shared in front of a laptop, the coffee machine, and in the old times, before pandemic, also at the air hockey table. A special thanks goes to the party girls and the salsa family, because they made my life here more Italian, real, and alive. As soon as I met you girls, Agnese et al., I realized, ho capito quanto non sarei stata da sola in questo viaggio. Siamo tutte PhD, siamo tutte ugualmente incasinate con i nostri problemi, ma grazie a voi siamo riuscite, o riesco quantomeno, a distrarmi, discutere e capire che non sono problemi abissali. Quindi ci ridiamo su, ci beviamo su e alla fine va tutto bene. Carl and the dance team instead introduced me to a new world, a new environment, as international and exciting as the academic one, but where people's backgrounds was way more varied. Thanks to you, the not so open-minded Italian has become the international loving person that I am now, which I would have probably missed if I hadn't come here. E alla fine, grazie a tutto il mio sistema di supporto, famiglia e amici che sono stati con me praticamente fin dall'inizio. Non importa dove siete, o se siete su o giù. Grazie a tutti quanti per continuare a essere con me. E Nazi, non ti ho dimenticato, solo che non ti posso dire quanto ti sono grata per l'aiuto che mi hai dato. Grazie. That's it.
Okay, thanks for these lovely words and speech. I try to understand small parts of the Italian. I hereby officially close this session. I warmly welcome all of the examiners and, of course, the audience, the promoter in particular, for all that has been performed. So enjoy your day, Jessica, Dr. Samogin, and I hope you can celebrate it together with your family or friends, whatever. And yeah, hopefully keep in touch. As I understood, you will be with us at our faculty, at our lovely faculty.
indeed.
So hope to see you in the future. Congrats again. You did a great job.
Thank you to you all, really.
Yeah, we would like to thank the chairman for his nice job.
Thank you.
Thanks, Stefan.
Congratulations.
Yes, congratulations, absolutely.
Thank you, professors.
Congratulations.
Thank you. I'm grateful to you all.
Very well done, Jessica.
Congratulations, Jessica.
Thank
Enjoy
you.
your day.
Thank you.
E grazie mille Elisa a te
Sì,
per essere stata con noi nella commissione. Magari un
Grazie a
giorno
voi.
riusciremo a invitarti a Leuven l'anno
Volen-
prossimo, direi, ma
Volentieri, speriamo
oppure
presto.
veniamo noi. Oppure veniamo noi.
Speriamo presto di poterci incontrare.
Speriamo.
speriamo. Grazie mille.
Thank you again. Bye.
Bye.
Congratulations, Dante, to you as well for your critical supervision.
Well, Stefan, it's the last project. It's certainly the one that Jessica liked most, and I also do. And that was possible because you are with us, working with us. So
I almost thought she was a perfect ambassador for the study of aging research at the end of her talk.
that's nice.
Or at
Well,
the
that's
end of
maybe
the
because I
discussion
like it so
section.
much.
Future. Okay.
Yeah, I liked it.
Bye, Elisa.
Jessica.
Bye.
Bye.
I was going to say, I hope you have a safe trip back home, but there is no trip, right?
There is no trip.
But again, hopefully we'll meet sometime in the future in
I hope so.
a
Soon.
Thanks a
non-digital
lot,
manner.
Jeroen.
Bye-bye.
Bye.
Thanks, Stefan.
Bye.
Jessica.
Thank you.
Bye.
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 Dio, ce l'ho fatta.
Dottoressa,
Aspetta, che non
questi
lo
non
posso
sono da parte nostra, ma li consegniamo lo stesso.
Questi non so.
C'è il bigliettino, puoi
Aspetta,
leggere il
aspetta,
bigliettino.
un attimo, chiudo Skype almeno. Aspetta. Zi, buona. Aspetta, anche il camera. Ah, ti è, ho imparato. Questi. Sicilia. Che ne so, è roba di Macintosh. Servizi. Aspetta. Allora. Grazie family. Grazie mamma, nonna, vabbè, ma poi so che ci siete anche papà, tata, eccetera. Giusto? La tua famiglia non... Famiglia e non... Vabbè, immagino tutti quanti. Non so come abbiate fatto a raggiungere le ragazze, cioè Gaia. Ok. Grazie mille, sono bellissimi. Grazie. Spero non vi ho annoiato, ecco. Posto? Ci vediamo stasera, famiglia. Bye bye.